no, if I forget that. Okay, I think we have covered like from the early days of Gregory Mendel to Watson and Crick, the famous paper, where we talked about how they were able to scoop uh, the great Linus Pauling, and and that the rest is history. So that was the most famous paper. But a few weeks later, they wrote an even more important paper that Jim Watson wasn't that enthusiastic about it, but Francis Crick was more so about the and I'm sure I did mention in the conclusion of the first paper, that was the most understated statement, that it has not escaped our attention that this specific pairing of A with T and G with C, uh, you know, may give us insight of genetic, uh, code. Okay, and the second paper's conclusion was, it has not escaped our attention that this specific pairing we have proposed immediately suggests a possible copying mechanism for genetic material. Okay, so that was, so that was one of the this things. So because they, obviously everybody knew that the human body was made of proteins, and that's where the action was. So, but nobody had any clue as to how this genetic information was translated. And so that's where you know, he was thinking on those lines, as early as 53 type of thing. So, okay. And this is just to refresh the A and T and the classic, that's his basic high school, or for the Brits, A-level biology, or maybe, just, I don't know at what level you learned this, Peter, but, this was, really like a high school or even a middle school biology, basically. So, and so this are, so the event started. So hopefully we'll cover, and these are just various ways we can represent DNA, instead of we can show you the first, before the 3 minutes, it's a chromosome, basically, we have 23 pairs. It's a long molecule with bases, okay, which are intertwined, okay. And, this is another way of representing, okay. And this is one way of just like a long sequence of units, now there is no order, it looks random, but it is interpreted by a specialized machinery that's in each cell in our body, okay. So basically it has the A and T bound together, and C and G, they are complementary strands, so this is again, and so this is, one can say in simple words, that chemistry is biology, or at the same time we can say that that is a sort of computer science also, because that has a error correcting mode, that has a copying mode, that has a specific order. So you can see that that's, okay. Now, if you see actually DNA, that does not look very impressive, I mean, partly grossness, but it looks like a snot, basically. It's a colorless goo, and this was 60 Minutes had one of the CRISPR researchers, and he had held up this little nondescript test tubes. That's CRISPR? Okay, so that was, so none of these, agents did look that impressive. So, but basically it's a code, of that, and with various chemical bonds, as we talked about last time. Okay, just to, in the precise order, that's the information. So that is, and I think, quite a few of us have taken John's his class, and and that's, and if you don't have a copy of his book, then next time he does the class, probably, it's a very nice little book that that has goes much more beyond what we are talking about, So, okay, so I'll put in a plug for John's book and his class when he does it next time. Okay, and the other two molecules, we just to go over the basic, for those of you who are not biologists, that these are, again, these are various nucleic acid with different bases, okay, and as to which came first, there is a debate on it, but I think, John may disagree with this, but, they, I know, a lot of the people who are into origin of life are beginning to believe that RNA was probably first. But again, we did a whole class on it, so I'm not going to get into that as to becomes like a chicken versus egg as to what came first, and people have various pet theories as to what came first, was to whether it was the membrane that came first or the RNA that came first, or so. But it's another story, so I won't dwell too much into it at this stage. So, and okay, now, and that's a hope, and people have been awarded Nobel Prizes for RNA research, including the mentor of Jennifer Doudna. Okay, and okay, so that's and RNA has been a very popular, chemical for the Nobel Prize. There have been at least 15 Nobel Prizes have been awarded for RNA work, different types, and we'll go into that as we go along, type of thing, until as recently as within last 5 years, that an Israeli woman scientist and an Asian Indian who is now in Britain, were awarded Nobel Prize. I want to say 2009 for determining the chemical structure of ribosome molecules, and he went on to become probably one of the only Asians or foreigners to become the 62nd president of the Royal Society, which is, when considering how old the Royal Society is, that's a pretty high honor, that thing. So, okay, and so, all right, and again, just to give you a cartoon diagram, these are the proteins are made of amino acids, which is not a nucleic acid, by just to clarify, and these are long chains, which can be extremely long, anywhere from few molecules to as many as thousands. Okay. So this is just okay, and the basic principle that along with this, Francis Crick, I should say somewhere around 1968 had 1958 had proposed, the way the information was transferred. And this idea came in with various other phage scientists who were working with, we'll get down to another, work of a couple of French scientists, and that came up with the concept of different types of RNA molecules that were, discovered around at that time. And this was the basic as to how now the two types of cells, bacteria, do not have nucleus, while, humans and mammals and higher animals have nucleus, and the DNA is kept like a, I give you the analogy of it's kept as grandma's recipe that is kept in the safe. And when the neighbor asks for the cake recipe, that you make a copy of it and it's sent out. Okay, and then she copies it with various ingredients in assemble. So this is called, the word that is used in basic biology is called transcription. Means it's made a copy, it's transcribed, and it is translated into, the material that is useful eventually. So these are various amino acid changes are linked together. And so basically, so this was an idea that Siddharth Mukherjee enlightens in his book. Okay, genes that regulate and encode message, that protein that regulate again. So it's a circle basically. So genotype determines the phenotype. But we know that basically that's not as straightforward equation as that. There's a huge influence of environment, various triggers that take place, and also some randomness type of thing, that even two women may have the same breast cancer gene, but not both of them, and they may even have a similar lifestyle for that matter. They may be both overweight, but one of them gets the cancer while other doesn't. So sometimes, just pure randomness may be playing a role as well. So there are but environment plays a huge role. And there is some interesting work that is done by, people who are doing work on a special type of RNA that we'll get to in further down, and it shows that, okay, that things were not as simple as we always thought. Okay. And now I think I won't show too many videos. There's a link here. So usually I think hopefully this will maybe one of the few ones that I'll show, because this is a nice central diagram from the famous prestigious place we talked about.
The DNA double helix contains two linear sequences of the letters A, C, G, and T, which carry coded instructions. Transcription of DNA begins with a bundle of factors assembling at the start of a gene to read off the information that will be needed to make a protein. The blue molecule is unzipping the double helix and copying one of the two strands. The yellow chain snaking out at the top is a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA letter by letter to copy the gene. At this point, the RNA needs to be edited before it can be translated into a protein. This editing process is called splicing, which involves removing the green non-coding regions called introns, leaving only the yellow protein-coding exons. Splicing begins with assembly of factors at the intron-exon borders, which act as beacons to guide small proteins to form a splicing machine called the spliceosome. The animation is showing this happening in real time. The spliceosome then brings the exons on either side of the intron very close together, ready to be cut. One end of the intron is cut and folded back on itself to join and form a loop. The spliceosome then cuts the RNA to release the loop and join the two exons together. The edited RNA and intron are released, and the spliceosome disassembles. This process is repeated for every intron in the RNA. Numerous spliceosomes remove all introns so that the edited RNA contains only exons, which are the complete instructions for the protein. Again, this is happening in real time. When the RNA copy is complete, it snakes out into the outer part of the cell. Then all the components of a molecular factory called a ribosome lock together around the RNA. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. Inside the ribosome, the RNA is pulled through like a tape. There are different transfer molecules for each of the 20 amino acids shown as small red tips. The code for each amino acid is read off the RNA 3 letters at a time and matched to 3 corresponding letters on the transfer molecules. The amino acid is added to the growing protein chain, and after a few seconds the protein starts to emerge from the ribosome. Ribosomes can make many proteins. It just depends what genetic message you feed into the RNA.
Okay, so that was, very, well done. Now, the word intron was coined in the 60s, by a Harvard geneticist, named Wally Gilbert, and it means intragenetic region. That's why it was called intron. So that was a part of non-coding, and a lot of this may have been because the DNA has lots of copies, and there are various areas that were initially thought to be useless or so-called, even the word junk DNA was used, but we're finding out that has a lot more significance than we realize. It's not all junk DNA, and we'll talk more about it when we talk after the completion of Human Genome Project. Okay, so I don't want to go ahead of myself. So that's the basic 3 steps of transcription: the initiation, elongation, and termination. And so this is the peptides are in the direction of messenger RNA, which is single-stranded. The transfer RNA is hairpin shape of about 80 nucleotides, and then and we'll talk more about the code, as to how there is a little bit of redundancy or wobble effect that is, okay. So this is how the machinery goes on, and it's amazing how fast this it takes on inside each and every cell. Okay, so, and this is basically just gives you how the whole thing is put together. You may have seen this slide earlier as to how we translate it from cellular biology to molecular biology. And just to refresh your memories, I put in the slide again as to how, it explains the whole, from double helix to, and it's wrapped around, because otherwise it would be extremely long. It would be almost like 2 meters long type of thing. So, and then it's, and at the end, there are just little caps which have a lot to do with aging process at the end of the chromosome. And that, we talked at length in this class on aging. Okay, so this was, just gives you a fresh idea about how the groundwork was set up for the start of molecular biology in the late 40s and early 50s. And we are seeing, the work is still accelerated dramatically. Now, one experiment that was done, probably I should say around in the 60s, that was done by 2 French geneticists named Monod and Jacob. And they also had an American who was a researcher with them named Pardee. And their famous paper was called Pajama paper. They took the first initials of PAGA in Pajama paper. So that's how it's been remembered in genetics literature. Okay, and they were studying, E. coli and E. coli, bacteria, which was the workhorse of all the geneticists earlier. And normally it relies on glucose. Okay, but when glucose is not available and a different sugar, lactose is available, that's when, so they studied how the bacteria would utilize lactose, and this is where they discovered, the actual mechanism is, gets into too much basic biochemistry, and I don't want to put people to sleep for it, but they said, how the genes are there, but as long as glucose is available, they kind of stay quiet, and they are so-called repressed, by an element called repressor protein, which is in part of the head. So it keeps these other genes, okay, because any work that requires energy, so it doesn't want to waste energy. And so, but if glucose is not available, then the repressor switch is removed. So sort of negative feedback mechanism that, and that is removed, and then the 3 genes are able to start utilizing lactose, which is available, finally the enzyme just creeps inside the cell and says, hey, you should wake up and start working. So this is very simplistic regulatory mechanism. So, and since they work in all 3 genes work in set cooperation, they labeled it as a operon or lactose operon. It's a unit of DNA that is under control of a single regulatory sequence that is upstream. Okay, all right. So this was one of the famous things. So they correlated this genetic with the reproduction of what happens at the metabolic level. So this is, this was one of the ways to put the information together as to how it was used. And some of these ideas were done by a lot of the phage scientists who work with, tiny virus-like particles called phages. I'll have more to say about later. And then the concept of messenger RNA were finalized at this stage. So in other words, the operon that the terminologist had, the famous geneticist Monet and Jacob proposed, it's a cluster of genes which are transcribed together to give a single molecule, and so it encodes multiple sequences at the end. So this was one of the famous concepts that was done. Okay, and then by this time, the biochemists were ahead. They already knew that there were 20 amino acids, but the geneticists did not have much of a clue as to how they were transcribed and how the genetic code But interestingly, they got a feedback from some physicists who are working outside the box, so to speak, and a very colorful Russian scientist named George Gamow, who had left during the Stalin era and was working in Washington. Okay, and he had written a letter to Francis Crick and Watson about proposing. Now obviously he had never, he hadn't read a lot of the biology research, and he was just, he used to come with. Initially, they thought, who is this eccentric guy? Now he had earlier work on nuclear physics, and the, because he was a foreigner, he could not work directly on the atomic project, but but he was a well-known atomic scientist in his own rights. So finally they realized, that that what he is saying makes some sense. So along with this, so they started, coming up with the ideas. These were just like a sort of thinkers club. They hadn't done much of the lab work. A lot of the physicists, were not interested in actually getting down and doing the bench work. But so this was, and because, there were 4 codes, of DNA, and as to how much, whether you would need 2, if you take 2, then you will not be able to come up with the, for 20 amino acids, you'll still be left with 16 pairs. So mathematically, it would not work. On the other hand, if they take a code of 4 letters, there will be too many. There'll be huge lots of, be like you'll be wasting, sort of energy type of thing. So this is when, people were coming up with various ideas type of thing, and as to how to do research that, no, obviously the physicists were out of their elements, and the geneticists were not that well versed in biochemistry. So that is where the trouble lied, so to speak. So, but it took, and so again, these were, some varieties of ideas. So you could see some of the famous names of the physicists, so they were, saying, okay, we'll try to solve it. They had formed an informal club called RNA Tie Club to solve the riddle of RNA structure and understand how the proteins work. But, they were not, exactly interested in doing the real hard laboratory work, so combination of hard drinking and science, so, and you can see George Gamow was right at the top, Richard Feynman, okay, Edward Teller. So these are all various nuclear physicists. These people were, physicists turned paid scientists, So this is, this was the idea type of thing. So again, so this, so the work was still left to the people who were really willing to do the solid experimentation. Actually, is that, with thought processes and theoretical physics could only go so far. And now Crick had, always regretted the idea that the word dogma was, wasn't quite his intention. So it actually was a hypothesis, so to speak, as to how the code was translated. So because basically, obviously, the physicists knew about energy, the life involved flow of energy, matter, information, and how the information was translated. So this is where the thing stood at that time. Okay. And we already talked about the Cambridge this thing. So this is the code was okay. And by this time, so and I already talked about how if there is one basis, then we only get 4 letters, type of thing. If you have 2 bases, you get 16. So you're still left with missing the code for others. If you make 3 bases, then you have 64. Okay, type thing. So, but it took a relatively unassuming scientist, named Marshall Nirenberg, who was originally from Florida, and he was working in the early days of NIH. Okay, and by this time, he had, they had radioactively tagged nucleotides. Okay, and he was able to tag the nucleotide U. Okay, and with this, with biochemical experiment, he was able to, discover that the amino acid was formed with, with the code of 3 chain of 3 nucleotides UUU. Okay, and they were lucky also that they came up with the amino acid that the first one was phenylalanine. So that was so it was a thing. And at that time in 1961 there was a biochemical congress. Now this is, you can see it's the height of the Cold War, but the congress was held in Moscow. And by this time, his paper was at the end of the day. He was relatively young, about 25 or 30-year-old scientist, and he was presenting paper at the end of the day. So most of the people were not, had already left, type of thing. And then the story goes that Watson realized the importance of this, and they asked him to present the paper again. And once everybody heard what they had accomplished, then all of a sudden, this is before email, telegrams started going to their office, their laboratories back in States. And there were some powerful chemists were well funded, named Salvador Luria at Rockefeller University and other universities across the United States. They started tagging the nucleotides, and the race to start code, to break the code for all 20 amino acids took place. And and this was around 68, 70. This is a guy who was born in British India and gradually made from UK back to United States, and he was at Wisconsin, and he synthesized a lot of the amino acids. So Corona, Haley, and Nirenberg were Nobel laureates for cracking the genetic code. I forget exactly what year was that. If John or Peter knows that, please chime in. So this was, and probably that was the first time I heard of University of Wisconsin. When I was 18 years old. Type of thing, in a place called. This was about the same time when the riots were also in the news. I think so. And this was the sign that you are great, Marshall Nirenberg. So that's the famous story as to how the code for proteins was synthesized and cracked. And we already talked about, you may recognize phenylalanine. We talked about, that this is, we talked about one of the inborn errors of metabolism that we had talked about, okay. And this was a physician who was interested in genetics, who had come up with the test to discover phenylalanine, which I told the story of that earlier as to how, how the test was devised. And he was MD, PhD, and he came up with what is known as Guthrie cards, and it became standardized test for newborn babies. It became accepted, and then there was a controversy. He was trying to give a contract to one of the pharmaceuticals, Ames Pharmaceuticals, and but his idea was that this test should be kept relatively cheap, so it is easily available to people with limited resources. But Ames wanted to keep the cost at $250 in those days. He wanted to charge $6, so he started his own company. And one of the defenders for Ames Pharmaceuticals was a senator that you may have known about, called Senator Birch. He was not a senator at that time, but he was a lawyer named Birch Bayh. The name may sound familiar. And in 1980, he introduced a Bayh-Dole bill, which allowed universities and small businesses to retain ownerships of inventions that were done with federal funding. So it was, so that's an interesting story about how the pharmaceutical and university collaboration was. And again, so this is again like a lot of tests in genetics, that it is not without controversy. As recently as 2012, in Ireland, controversy emerged as to number of hospitals retained these test cards. So they had a DNA database with over 1 million samples from 1984, and that was done without the consent or notification of parents. So we had talked, I think, in the first class as to about how this data can be used against you, and if the police have access to it, it really invades your privacy to your data as to who really owns it. So this again, so at least in Ireland, this resulted in a 10 real rolling destruction cycle. So again, so the data can come back and haunt you type of thing. So, and this test, we told the story, it was done with ferric chloride. The test was actually devised in Norway. So, but that's an interesting story about as to how science and pharmaceuticals intertwined, along with concern for privacy. Okay. Now, again, with the discovery of genetics, there was the enthusiasm took place that anything that you win, people had this big hopes that any illness we'd be able to find a gene for that. Okay. And again, so this was the earlier work that was done for monogenic disease, where there is only one abnormality, the classic example being sickle cell that we talked at length about it, and there is, a nation database maintained called Mendelian basis of molecular diseases, that's maintained by NCBI. And there are about 7,000 diseases, just to put it in perspective. So this is their homepage title. So now that is very legitimate, but at the same time, this is a British slide, but you can see that how there is a gene for that. So anything that you'd wish, you can find. Okay, starting from the hype that goes on, as to how genes that take you good at taking risk, happiness gene. I mean, this is I don't know whether to laugh or cry. So make you scare you out of your mind, that thing. So this is, and even the other newspapers, this is a scientific, even the prestigious Scientific American was not be above and beyond this. Now obviously, this is an older slide. That this is schizophrenia is a lot more complicated than just a gene to say. So this is, and then I should say somewhere in the 80s or 90s, there was a controversy about something called a gay gene, So that was one way, but it didn't pan out when people tried to duplicate this, and I think to put the science in perspective, there are some areas in brain, which are different in people with in homosexuals. So that was more that has more solid anatomical basis than genetic basis, So and I think I'm trying to remember, it's called bad nucleus, stria bad nucleus of somebody. So that is, I forget the actual area's name. So, but the hype that just went on was unreal. And I think in my other class you would see, like how there are genes for just about everything that the single, it's become like a curiosity change. Okay. Now again, that we talked about, the code that we talked about, that is a rather simplistic ways, because in eukaryotes, because of duplication of gene and something called copy number variations, the genes are a lot more complicated. So you can see that they are not just all lined up together, because there is a little gap in between, that is called intron, that I explained to you a little while ago. Okay. So, and this is gap. So in other words, like the word genome is not written just like in cartoonists. It's G dash dash NO dash dash ME. So it has like a start and stop and cutting out portions, And so this may be, this is how, works. And, that's why you don't get your liver cell in your kidneys and kidney cell in your brain, So, okay. So there is like you can say there's a go mechanism, okay, which starts the sequencing. There's a stop mechanism. Okay, and in between there are areas which are useless. So before it can be translated, there are signals that need to be cut out into a region, So there's like a proofreading, and you have to decide when to turn this on. So you don't start making insulin in your brain, and as to what time it should start. In other words, you don't start making sperms when you're, or testosterone, before your puberty type of thing. So that would be, it would be bad. So this is, how the sequence, and there are various fancy names for it, but that we'll leave it to the real molecular biologists. I can give you the name, but so in the beginning, this is a statement, by Richard Dawkins. In the beginning was simplicity. And these are the fancy names, for people who really want to know type of thing, introns. Okay, so this is, we talked about instead of the simple code that we talk about exons and intron, start and stop codon. And, so it gets a lot more complicated than you care to know at this stage, so, okay. All right, and again, and this was a very gradual discoveries that took place, that as far back as, in 1959, people discovered how RNA is synthesized, as to varieties of sequences. A lot of the work was done in yeast and E. coli, okay, and the transfer RNA was discovered, the sequence was discovered in 65. And so it was a gradual process as to how the science progressed, So, and by this time, finally the genetic code was being, finalized, Okay, and then okay, let me go back to the genetic code. That's a little bit interesting type of thing. So if I can go back to, okay, yeah, I didn't elaborate, type of thing. So this was, and so that, and you can see that how, methionine was the first code. There's only one 3-letter code for that, while some of the other amino acids have redundancy. So they have varieties of codes. So this is, and the first 2 letters are significant. The last letter can be, so that's something called the wobble or the redundancy in the code. So we can see that there is, they have a permutation combination of up to 64 of 3 letters, okay, but there are only 20 amino acids. So there is, and there is something called the start codon and stop codon. So these are the stop codons type of thing. And the start codon is AUG. So this is, this is how the genetic code is. Why that happened that way, that nobody has any scientific explanation for it. But that was a little bit of more details on that, that was worth mentioning. All right, stop me if you have anybody has any questions or comments. Oh, I'm sorry, I should have been looking at. Oh, okay, yeah. Well, yeah, Peter's comment was that this occurs in a very constrained environment. That's what makes it possible when, okay. Yes, the curves happening. I have some, for sake of time, I'm not going to show all the other videos from Cold Spring Harbor, but there is one thing about the histone wrapping, I can send that to you earlier as to how the DNA is wrapped around the histone. And I may have some later on type of thing if we have time. Okay, Patricia has a question. Is there a gene for celiac? Okay. Oh, it's more of an autoimmune disease, so it's a lot more complicated than they think. There's no isolated this thing. It's a, the genes may play a role, but more often your microbiome plays a bigger role for celiac. So when I don't know if you were in the medicine class or not, but when we talked about how your microbiota and the gut lining has a lot more to do with celiac disease. So one may have a genetic tendency, but your diet and autoimmunity, plays a bigger role, in celiac etiology. Okay, so hopefully that gives you some thing as to if we are.
On Peter's comment about the constrained environment, why does the constrained environment make it possible?
Okay, Peter can answer that if he wants to jump in.
Sure. It's really just the thermodynamics. If you imagine 2 things needing to bump into each other to actually do something,
Okay.
then if things are happening in that big swirly space, they'd never bump into each other. So what happens is you end up with really a compartment in the cell where all the bits are readily available, and the chances of them bumping into each other are quite high. So that enables it to go fast and happen in our lifetimes. Otherwise, it just simply wouldn't happen. It would be too remote a possibility.
Okay, thank you.
But it would be completely impossible to illustrate. So the animations are just wonderful.
And actually, I'm not going to get into the cell thing, but in the intracellular networks, that it is so complicated with microtubules and other things, and the membrane and the cell-to-cell connectivity. That's an altogether different. If you see the cell signaling chart, I mean, that's just amazing how people can figure out how many enzymes, are playing a role in just starting from the entry into the cell, from the membrane level to the ultimate product that is. So the intracellular network, cell signaling is a whole together different class itself. So, but it's almost like a more than a graduate level class. So I don't want to, but if I could show you sometimes, I think I'll try to bring a chart of the intracellular networks. There are some of the biopharmaceuticals send the chart about, it looks a lot worse than the, metro map of London subway. It's a lot more complicated even than that. So okay. And as to how the proximity of cell-to-cell networks, and there is a huge role that microtubules also play a role, and that may be one of the explanations for how anesthesia works also. And so those are even smaller structures than the rest things. And I think that's why I think I'm just going through the book by Siddharth Mukherjee about the song of the cell that I mentioned last week. So unfortunately I started reading from the back, so I think I haven't got to that part yet. But but that may be something somebody who's interested as to how the cell itself works. So it's a whole village basically, not just an isolated. It doesn't live in isolation. And we talked a little bit about when we talked in many medical school class as to how they don't work in collaboration. It's like so. Okay, and when I talk about I'll have one or two slides about stem cells. So that's where I'll get into it a little bit, how the fibroblast and others play a role. Okay, now we talked about mutations. We already talked about the simple mutation of the one amino acids that we talked about. Okay, of this thing. But there are other mutations. There are some that are silent, which don't cause any problem. Say, okay, well, just a simple and there are substitution, which the meaning is changed. And there are deletions, means that the part is eliminated. And there are insertions that take place. That just totally. You can see how the sentence doesn't make any sense, basically. So this just gives you some idea about the varieties of mutations. So in other words, there's mutation and there's mutation, so to speak. So and then again, so there are two types of mutations that takes place. If mutation takes place in your germline, that is in your sperm or ova, then your whole generation, just like we gave you the example of Huntington's disease, okay, or type of thing, where just one founding member, can condemn the next generations for next 500 years, basically, and you can't do much about it. On the other hand, if it occurs in one isolated cell, so-called quote unquote somatic cell, then it's once you died, or it is cured, then there is not much further effect on future generations. So, and we talked about how some things are dominant, with the examples we gave was Huntington's, and then the sex-linked, which is the hemophilia that we give examples. Okay, and again, so these are just the mutations in the genetic code. And then we also talked about the chromosomal abnormalities, how the chromosomes can be duplicated. We gave you the 4 examples of duplication, which is the trisomy 21, i.e. Down syndrome, translocation, okay, insertion and deletion, so which are I gave you the first example. And then there are other types of mutations take place. They are called copy number variations, which are this triple code can keep getting repeated and amplified type of thing. And that occurs in Huntington's, that the sequence is repeated multiple times, so that, so in some cases, the copy number variation can be beneficial for multiplication of human for genome and coming up with new functions. So that is one of the big mechanisms how the genes, have copied each other. So the human genome has been copied since the evolutionary times about 7 or 8 times, and there are certain sequences that are repeated about thousands and thousands of times, which we still don't understand what is the significance of that. Okay, and so, and again, so a lot of these things, and at the same time, so these mutations occur primarily due to environment, chemicals, spontaneous, so the varieties of causes, and a lot of the impetus was started after seeing the Hiroshima, and that may be one of the reasons why, the the government was interested in funding the research for doing the Human Genome Project, so, and so a Department of Energy was more interested in getting this started than NIH for funding the research, because they were interested in the effect of radiation that was taking place. And people learned that a lot after the Hiroshima as to how the finally, people initially, obviously, thousands of people died initially, and people died in initial radiation sickness within first week or so. And the third wave of killing that occurred from Hiroshima was after about 6 to 8 months, when their bone marrow failed. Okay, and they started bleeding. Some of them got cancer. Their bone marrow stopped manufacturing. So that was a third thing. So that was one of the big research for Department of Energy to determine the genetics, and that led to the funding. But more on that a little later as to how the funding came about. Okay. Again, so how genes are complicated, as we said. So how this is like simple evolution of color vision. As to how evolution of color vision initially, we were before in the evolution when they were aquatic animal only, they were not, they were able to detect only a blue light, and then later on the gene got duplicated and the color vision evolved with the ability to detect red and green, and that was on X chromosome. Okay, that is why if you have mutation in that, that's why the color blindness is much more common in men, than in women, type of thing. So, and again, this gene has been duplicated in two, has appeared in 2 separate times in the New World monkeys. I'm sorry, let me get, okay. Oh, they have a dichromatic vision, while some of the howler monk females have trichromatic vision. Okay? So this, so the gene has been has appeared and reappeared, twice on the evolutionary scale. Okay, and similarly, that at the time also. So this is the appearance of the location of the gene as to where, at what type in the evolutionary scale it appeared. And in terms of hemoglobin, there is a time relationship as to how the genes, turn on and off, so to speak. Okay, obviously with the placental birth, maternal and fetal bloods does not mix, and but at the same time, the fetus needs to get the oxygen from a mother. Okay, so the globin protein, which is the basic protein with higher affinity, has evolved, in the earlier phase of life. Okay, and then it gradually disappears as the child grows. After birth, it disappears, so to speak, and because the fetal hemoglobin has a higher oxygen capacity, and after birth, that gene is particularly turned off, basically. Okay, and so and some of the lambda gene is basically created by gene duplication of fetal hemoglobin. Okay, so these are the various patterns of evolution that have taken place based on when the need arose, as to with the ability to perceive color gave an advantage on an evolutionary basis to the monkey, so they could get more energy from eating calorie-rich fruit. The same way, when the before birth, a child needs more oxygen. That's when the fetal hemoglobin gene evolved. Okay, so it's an exquisitely timed, relationship that occurred as to, it's like an orchestra as to which section would play the horns versus, violins would play. So it's a finely timed orchestra that takes place in your human in genome of all species, not just humans. Okay, and so patterns this is show that this region. So this is the protein diagram of the hemoglobin, basically. So this region is most important region. So there are the varieties of chains that are, that's more for the biochemistry. Again, when we talked about the 7,000 genes, that takes place, and unfortunately, in a relatively small population that tends to marry amongst themselves, but some of the diseases are a lot more common, and especially in Ashkenazi Jews, some of the diseases were, a lot more common in that particular subgroup of ethnicity, and one of the rabbis, had like 4, 5, or 6 children who had died of Tay-Sachs disease. So he had started the program, that is the Yiddish name called Dor Yeshorim, okay, for screening, for a test before a couple decides to get married, and especially and at least before they decide to have children, to screen. And on the East Coast, they were able to eliminate the disease even before all the major genetic screen testing, but the genome analysis became available. So, and they've had a very successful outcome, basically. So, I mean, they've hardly had they've not missed any, and so they have been able to avoid this life-threatening disease, which is a childhood disease where the mental retardation and various physical abnormalities occur. And then it has been expanded further, this is a non-profit saliva test, type of thing. So, and at the same time, they're able to maintain privacy. On the other side, in other religions, this was a report just this week in New England Journal, okay, that this was a report from Canada that was a Pakistani family. Now, unfortunately, in Pakistani and some of the other, and also Saudi cultures, they tend to marry their first cousins sometimes, and that's a big problem for the NHS in UK, okay. But this was a family in Canada that they've had early onset, they had 2 children with mental retardation and something called lysosome storage diseases, which are very rare. And they were able to, for the 3rd child, they were able to diagnose it, in utero, and were able to do in utero enzyme replacement therapy. Okay. And this is, for Pompe's disease, an extremely rare genetic metabolic disease, which is, you always, for people with medical background, this is always, you see it on the last page of Harrison's textbook. Okay. And they were able to treat it in utero with enzyme replacement therapy. So this was I just put this in this morning because it was in the New England Journal within last 2 weeks, I should say. Okay. And so they were able to successfully treat, in utero enzyme replacement. So this is, but that is, that's a big this thing about how common, and the NHS, as I mentioned, has a huge problem in the immigrant Islamic population, primarily from Pakistan, and also Saudi Arabia has, they're richer, so they're able to do a lot more screening now, and so they're spending tons of money on genetic testing. So the 2 different ethnic communities have different approaches and different outcomes with serious genetic diseases. Okay. Now, at the same time, the other enzyme research was going on, and this is, the various the people who are doing the harder work. This is the various enzymes were being discovered, because the enzymes are the workhorse of all these things. The DNA and all stuff is there, but it's the enzymes that do the hard work, type of thing. So, and this was the earliest discovery of various enzymes using E. coli, and this was done in the 60s, I think. So, and this was, and it's interesting to see that this report was declined by the Journal of Biological Chemistry. So your biochemists are pretty tough, And this work went on to get the Nobel Prize, by the way, for Arthur Kornberg, Okay. So amongst the critical comment, it is very doubtful that the authors are retired to speak on enzymatic synthesis of DNA. Okay. I mean, it's an interesting to read the comments. And when I read that, I thought this was, so I'm sure, Arthur Kornberg had so this was, so he, I don't know where he was at, when he did the work, but I think he was at Stanford then. And again, the concept is that the genes, provide the message, it causes gives the protein, and all of this work is, communicated by various enzymes, So you can see, like in cosmology, in the medieval cosmology, there is a joke as to what is underneath everything, all the evolution is turtles all the way. So one can say that this is, still is genes all the way in biology, So, okay, so we have come from a long way from determining dissection and gross anatomy to microscopic anatomy to finally, finally the DNA takes the bow, so to speak. So that's where we're finally getting down to the reductionist approach at the bottom, basically. So. Okay, so regulation, replication, and recombinations, all the, okay. And this is, it's an interesting story about, these are the two, it's just like the royalty of Nobel family, it's just similar to the Curies, the Kornberg family, Arthur Kornberg, a Nobel, as a medical student, he has diagnosed his own disease. This is a rare liver enzyme disease, okay, that is called Gilbert's disease, And that's when he was interested in genetics. I think he was at Rochester or, on the East Coast, And then he went on to do work at NIH and then at Stanford, about the discovery of DNA, of polymerase enzymes. Okay, and and so by this time, there were various restriction enzymes that were available, and so they were able to cut. And these are various enzymes that were cut by this E. coli enzyme, okay, restriction enzyme E. coli, that's how it is named, paste, but there is another enzyme called ligase, then it was copied with the tiny virus particles. So this is, this was one of the ways they would work with E. coli. And this was his son, Roger Kornberg, and who also was a Nobel laureate. So he had discovered some other enzyme, which are RNA polymerase, okay. And this is, we'll talk about histones later on when we talk about epigenetics later. So these are the two Kornbergs, families, where, and as other brother Thomas Kornberg had discovered other enzymes. So you can just say they're like the biochemical royalty, so to speak. Okay. And at the same time, we already talked about the central dogma. Okay. But on the other front, some interesting work was. Okay, just to digress, back in 1910, a scientist named Rouse had discovered a virus in Rouse sarcoma in chickens. So this was left, and it was left at that the viruses were thought to be causing, the tumor in chickens. So that was called Rouse sarcoma virus, So okay, so that was where it was left at. Okay, and then. And with the research that was being done by the two guys named Baltimore and Temin, okay, they were discovering observation on phages, and this was the cells that were transformed by the Rouse sarcoma virus led him to the conclusion that there were viruses that were working, causing their effect through DNA intermediate, which he called provirus, and so this actually was violated the central dogma, okay, and this was again, everybody was initially very skeptical about his work, and Howard Temin was again at University of Wisconsin, and and when he had presented his data, at the same time, it happens quite often in science that the same work was duplicated by a person who was already an assistant professor at MIT named Baltimore, okay. So both of them had come to the same conclusion that the retroviruses were working, to violate the central dogma. Okay, and so this was, and instead of the DNA to RNA to protein that I just, repeated myself in numerous times, the viruses were going backwards, so to speak. It's like how RNA were affecting the DNA, and the RNA viruses, and then creating this thing. So this was the principle. Was being violated, okay, and this is, this is what viruses look like, so they are like a small package of RNA or DNA code, that's obviously with the COVID, everybody's familiar with that. There can be different types of viruses material, RNA and DNA, and there are 7 different classes of viruses, single strand, double strand, okay, and type so there are 7 different, it's called a virus. It's labeled actually Baltimore classification. So, and the HIV virus is the retrovirus, by the way, the AIDS virus. Okay. So this was, the big, this thing. So, and this again was paradigm shift in genetics, how the DNA from RNA. So these are some of the collage of nature guidelines as to how reverse transcriptase, so this was about 50 years ago. There was discovered by Baltimore and Temin, who were who were Nobel winners for the discovery of the retroviruses and the reverse transcriptase. So again, so the central dogma reverse. So this was the highlight in genetics. So there are a variety of science magazines. Where they had a field day with this. And again, by this time, as we talked about in the class on cancer in mini medicine, and the Lasker Foundation had a big public information campaign, and this was the early days of chemotherapy that we talked about, with leukemia, with Sidney Farber and Nixon. This was a big advertisement that took place. And Mr. Nixon, you can cure cancer, and this was Howard Temin, who was on the cover of Newsweek magazine, The War Against Cancer, looking at his slide, okay. And this was and that discovered the SRC, the sarcoma gene, okay. And so this again, and by the same time, this was 73 with Roe versus Wade, okay. And so this, and this again, it was I had told the story at probably the mini medicine class, but this is worth repeating before we take a break. The roots of this National Cancer Act can be traced to a small home in Watertown, Wisconsin, in the early 1900s, when a girl named Mary, who was different, and who saw her laundress, Mrs. Belter, who had breast cancer. So that stuck in her mind. And then she married, a big business executive and went on her own to go to Vassar. And then after he passed away, she made her cause to raise fund for treating cancer and started a huge, campaign to put the government's resources behind research. And the budget for cancer went on from a few $1 million to almost a couple of $100 million in Nixon's time type thing. So that was one of the things about Mr. Nixon, you can cure cancer. And Nixon, listened to the public demand, and that was when the NCI got the huge funding rise. Okay, I think we're coming up to 2:00, so we ought to take a few minutes break and then continue our tale as we go along. I will pause the recording for a while. I think resume recording. Okay, so this was the start of the, and in a way that you can see that the untimely or timely discovery of this virus and cancer gene, kind of through the research on cancer, on a wrong path for a long time type of thing. So now this is, I don't know how to tell this story. Let me see that how. Okay, this is an interesting story. I thought this was more of a human interest type of thing. So this was Ernest Henkin was an English bacteriologist, and there must be something about the English as to how they were polymaths. So he was not only just bacteriologist, but also an aeronautical theorist, naturalist, and he was working mostly in India, and he was studying malaria, cholera, and other diseases, and he had heard the myth that people who were, drinking the water of because cholera was rampant in those days in the subcontinent, but that there was something in the Ganges and the Yamuna River water, the so-called holy rivers, that those people who used that water, they had less cholera outbreak, was self-terminated, so to speak. So he was suspicious that there was something in the water that was protecting these people, a type of thing. So, and what it was, he couldn't put his finger on. But then subsequently, with filtering technology, a Frenchman named Felix, I'm not going to mispronounce it, Herel. Okay, he had filtered this and he had discovered that there were tiny particles, viruses type of thing, so those were attacking the bacteria. And because they ate the bacteria, that's how the name phage was given. So those were tiny particles type of thing. So that was the discovery of the phage, and that became the workhorse of the biochemists, and this was about 40, 50 years later, and then the big who is who branched out from physics to biology based on the inspiration of Schrodinger's book, a guy named Max Delbruck, Salvador Luria, Sidney Brenner, Watson, so they were all working with phage, so this was called the phage groups because they were easy to study, they multiplied quickly. And there was a big phage group that was at Caltech and Cold Spring Harbor, and that was, they became the big vectors, and so they were easier vectors, and a lot of the work was done in Georgia, which is not Georgia, Atlanta, but in Russia, former Soviet Union, Russia. So they're big into the phage work, and they're using it as a therapy for bacterial diseases before the discovery of penicillin. But with the discovery of penicillin, and primarily because it was communist, it fell out by the wayside in the Western culture. Okay, and this was one of the phages, was one of the first one that was the DNA was synthesized. A 5X174 phage, and that genome was determined, by a English geneticist, crystallographer, whose name we'll get to later. And this is what, the phages would look like under electron microscopy. So this was an interesting story as to how the old myth of the holy water being protected. So there was some phages inside this that protected against the cholera type of thing. So this encompassed 3 continents, basically. So this was global science truly at work, so the viruses that infect cells. Okay, so so they're all very similar, but basically, so you can just see 3 different terminology. Viruses are they're not all bad guys, but at least, to us, they are bad guys. They can infect cells. Phages are that infect bacteria, and plasmids are the kind of peace-loving, living independently inside the cell, So these are the 3 words, that are used. So, but this was an interesting link that. Okay, now I think this is I may have told the story of earlier about this book about scientist race to save her husband from a deep. Okay, now I think this is a long video, and I think I'll just send the link type of thing, But okay. Okay, and this was the story is this, that in March of 2016, a PhD psychiatry professor from UC San Diego named Tom Patterson became sick while he was visiting Egypt, and he had gallbladder and pancreatitis, and he was, then he was flown to Germany, and the bug was multi-drug resistant bacteria, and it was, he was dying from organ failure, and at that time his wife, who was a PhD epidemiology scientist, she had heard that there was, as a last Hail Mary pass, she thought that phages might be beneficial. And then she was with the research of UC San Diego and the Military Research Institute in Maryland. They were able to make a phage cocktail for his particular bug. And it was able to save his life, and she went on to write a book about it, okay. And this is a book by her name is Stephanie Straddy, okay, and Tom Peterson, so Perfect Predator. So this just tells about the story of the, about the clinicians who looked after him at UC San Diego. But so phage therapy may be making a comeback, but that has to be individualized depending upon so it's almost absolutely precision medicine, to put it mildly.
And I saw.
I'm sorry. Oh no, that was starting type of thing. So And so this is, just talks about, how the gene editing, this thing. So, and so the phages, okay, and we talked about how this led to this early days of gene editing, which where the plasmids were used, and bacteria multiply in a different ways, how they can exchange. This is something called, we referred to earlier about a horizontal gene transfer that takes place. They also can multiply by a method called conjugation, okay, and the varieties of ways bacteria can multiply, type of thing. And this is an electron microscope of plasmid DNA. This is how it just looks like a loop, of a basically. So that's basically a genetic material that is free lying, okay. And then again, when we talked about, and that the biochemists have known about various restriction enzymes that are used to, work with, from E. coli. That is the bacterial defense system against viruses. So there are like, huge numbers that have been around, and synthesized, and the scientists have known about it for a long time. And there are as many as like 7,000 so-called restriction enzymes. And the one that has been the workhorse of this is called ACOR1, and that is devised from E. coli, okay, restriction enzyme number 1. So that was the ACORI1, that was the enzymes. Now that is of important in the early days of gene editing. That's why I'm making this thing about it, okay? So, and these are, this is one way that these are the enzymes that help the bacteria restrict the viral growth. And this is very specific. This only works when they recognize a specific sequence of genetic code, like, for example, GATTCC, okay? All right, so this is, so that the sequence is very important as to how the evolution, and the word plasmid was coined, back in 1952, and it describes what is called extra chromosomal hereditary material, that was, that was discovered at that time, and so this is, so the war between evolutionary things just goes on. The viruses are attacking bacteria, and also, so this is where the bacteria have evolved the defense mechanism. And again, so this is important, when we talked about CRISPR, because that is one of, again, the defense mechanism that has evolved. And the sequence GATTCC, that was the 1997 movie Gattaca. So that is the sequence that, that was a movie that was one of the early movies about gene selection, as to how the desirables and non-desirables, the people who are qualified to go to the outer space and those who are not qualified. So it's a new movie. So I think I've mentioned that in the past, so it's starring Charlie Thompson, and I forget who the actor was, So, but it's a fun movie to see about how genetics, selection can backfire, and that's how the privacy becomes an important factor. Okay, and so this is, was the start of gene editing, type of thing that was done, as to various cells were assembled with plasmid, and based on that, the bacteria was re-engineered. And so this was the start of genetic engineering that took place. And there are some interesting stories about that we'll come to in the next couple of minutes. So let me just jump forward. Okay, so from plasmid to virus, this was the earlier work that people were working with plasmid, as I mentioned. And this is the man named Paul Berg. He was at Stanford, and he worked with a plasmid simian virus 40, SV40, that was on his license plate. And so this was, again, can cause cancer in hamster, but does not harm humans, okay. And so this was used as a tool for gene editing, okay. And so, okay, and let me go back to this slide, okay. And how, say, this is the plasmid that was inside the cell. It was, okay, it was reassembled, and the plasmid was able to re-engineer cut the bacteria of the chromosome in a specific sequence. Okay, so that's how the genetic engineering got its start. Okay, and this was, one can say that this was the classic revolution that took place, and this was the paper that started the whole this thing. Now, before this, one of his grad student was going to do an experiment of introducing this particular plasmid gene, which again, as I explained earlier, that can cause cancer in hamster. They were going to put it in E. coli, okay? And this experiment, so she presented this idea of this paper, at one of the East Coast conferences, and that almost, frightened one of the other scientists who was at the meeting in Massachusetts named Bob Paula, okay? And he contacted Berg as to why you're doing this particular experiment, because if this E. coli virus escaped the lab and infected, humans, that could be really dangerous, okay? And so that really first time it caused the concerns that that type of biological experiments are not exactly safe, and just because one can do it does not mean we should do it. And Bob Burke, initially just didn't think much about it, but after some deliberation on his part, he actually stopped the experiment of putting this potential cancer-causing virus in a very common garden variety bacteria, i.e. E. coli. But, and he was not that particularly interested in that experiment. But, they did also the reverse experiment that they were able to do, as to put the same virus into a mammalian cell, okay? And again, by that time, as all of us, are old enough to remember this particular book. So there was enough scare that was going on. And the concern that was on the part of some scientists was very legitimate, that this, that the experiment can backfire and cause a problem. Okay, so now, and this again, unlike people in physics community, who actually were, feeling, after the Hiroshima bombing, they were actually were having, guilt or something, and at least several physicists resigned, but quite a few of them continued. But unlike physicists, the genetics community, decided to put a moratorium on themselves. And Paul Berg actually, after a consensus with various other people who are working on the field, published a letter in the two prestigious magazines, Science and Nature, to putting a moratorium on recombinant research, with the possible, danger to the people at large. Okay, and so, and this by this time, so this is, one can say that the geneticists, were for the first time, they decided to police themselves and took the initiative to avoid experiments that have potential hazards. And at the same time, this was at this time the protests with Vietnam were going on. And as you can see, when the earlier experiments were done, Johnson had congratulated that they were able to, cut the DNA and do this thing. So within 10 years, the thinking in the society in general had changed dramatically. And as to people who are, instead of showing that as human beings we could do anything that's possible or can be doable, should be done, to the climate had changed dramatically. And for whatever reasons, at this time, Nixon had abolished the Office of Science and Technology, okay, for some reasons that are not known to me, type of thing. And so, and the scientists were also worried that there may be more stricter laws that may be coming up, and people who presented this data, and the Europeans were ahead that when this work was presented in Europe, a lot of the younger scientists, who were more concerned with society in general, while questioning the validity and usefulness of this type of work. So there was a global debate that took place in all the three major Western countries. I mean, like in UK, obviously a committee was appointed to investigate this, and in US also, a moratorium was considered. In France, there were protests. Same way in Germany, there were protests. And so this was, the climate, the social climate had a huge impact. And so, and based on this, okay, well, actually, okay, so this, I should have, my slides were out of sequence. So then at this time, there was a famous conference that was held in Asilomar in Montego Bay. And this is where, and this was an old symbol that was biohazard, that was first proposed in 65, and that was reused for these purposes. And after this conference of various who is who of genetics, several lawyers, several ethicists, the protocol was devised as to biosafety level, as to BSL-1, biosafety level 1, 2, and we've all seen with wearing the hazard and personal protection suit during virus, epidemic. As you can see, the level 1, E. coli, basically, level 2, influenza, HIV, which, and level 3, tuberculosis, Ebola. So you can see, well, how these are, and this is a rather complicated slide for useful for people with research. But explained simply, okay, level 1, don't eat it. Okay, yeah, you don't want to eat E. coli. Number 2, don't touch it. Yep, you don't want to touch, have be contaminated with HIV blood, okay, or tuberculosis, don't breathe it. Last but not the least, Ebola, don't do it here. Do it in a high security, with through protected ventilation type of laboratory, so biosafety hazard. So this is how they set protocol. And at the same time, the political climate was such that this conference was, again, the public itself did not have much input in it. There were reporters there, but again, they did not address some of the issues, but they were not germane at that time because gene editing for humans was still far into the future. Okay, and again, they did not discuss, the bioweapons type of thing, which some of the weaponizing some of the bacteria research was going on in Soviet Union and also in the United States, in under the military establishment. So they did not discuss some of those things, but at least they put some resemblance of societal context. And at the same time, there were protests on the East Coast, primarily in Cambridge when Harvard was opening a lab for recombinant DNA research. A mayor of Cambridge, who was a savvy politician named guy named Alfred Bellucci, had hearing and was able to actually stop Harvard from building a lab in Cambridge. So they ended up eventually building it further out in the other areas. Okay, so there were a huge public hearing and huge protests, primarily from a lot of students and younger scientists and public at large. And there are some other people, who are, savvy politicians and who have made a good knowledge out of it. So again, we can realize that biochemistry is not mathematics and it can be a lot messy. And this we already talked about, how the virus and interactions. Okay. And again, the concern around the same time was occurring with, various prion diseases that was taking place. And okay, and around the same time, the discovery of Kuru, which was due to even a particle that were even smaller than viruses, which are just misfolded proteins that was taking place. And so there are things that were a lot smaller that have been implicated in human diseases, And the term prions actually refers to a very small pathogenic agent that can, which is misfolded protein, which we talked about, type of thing, because the proteins are the workhorse of all the biochemical reactions, And so, and these are not well understood, but later on have been implicated in the mad cow disease as well in UK. And again, so these are pretty, untreatable diseases, and the one rare disease that again, which much more common in Ashkenazi Jewish community, which is the Creutzfeldt-Jakob disease, okay, and a Kuru, which was reported by the people who are working in Papua New Guinea. Okay. And these are some of the pictures of the protests in Cambridge as to how it became like a political theater. The hearing were held and Cambridge, in the public limelight. And this is one of the cartoons as to how you could have a P4 lab, but the bacteria can still escape, Okay, so there were varieties of guidelines, and in UK, a Society for Social Responsibility was established, okay, and they also had published, various pamphlets as to the social impact of biology, So the controversy went on between the old guard of the biochemistry and the geneticists established versus the younger people who are more socially conscious. Okay. And again, this is, I think I'm not going to show this because this is a 13-minute, this is like, remember, this is the days of hippie era, okay, and in the heydays of San Francisco. So the biochemists at Stanford Chemistry had fun with this. And this is.
Bound they were in the glorious sun by initiator factor 1. Initiator factor 2 went searching for tRNA, who bore the flaccid amino acid.
As I said, this is a long.
Then, as the uffish bonds were formed, the 50S ribosome untamed came whiffling through the tulgey grass and burbled as it came.
50S ribosome! 50S ribosome!
Okay, so this reminds you of the musical Hair, basically. So it's a long video, so if you want to waste time, you can watch it, type of thing. Or if you want to feel nostalgic about the 70s, So that's the link, So, but it's too long for us to play the whole thing. But ultimately, a lot of these things, actually gradually change, when and with the dawn of biotechnology and when people started to commercialize, some of these restrictions, were fallen by the little wayside because it became a big business. Okay, and that's an interesting story that we still have time to cover, type of thing. And this was genetic engineering. So this was two people, as I mentioned earlier, that Berg was working, with plasmid and trying to work with the enteroplasmid and modify the DNA inside the cell. Okay, and one of the other person who was working in his lab, named Herbert Boyer. Okay, and at the same time, another scientist from the East Coast named Stanley Cohen was working with plasmid. So, and we said this were the various cutting enzymes were available, to cut the DNA in a precisely same location. Okay. So, they were together at a meeting in at Hawaii, and they decided to work that they could modify the DNA and cut the DNA and insert a precise gene with the plasmid at the same place. So they came up trying to put the two interests together and to be able to edit and modify the DNA sequence inside the E. coli. And then once it is cut and the gene is inserted inside the E. coli, because remember the E. coli gene is eukaryote, so that's just like a big circle. You don't have to get inside the nucleus. So this was one way to edit and insert a desired gene. And they were working with antibiotic resistance. So those were the early experiments that were done, that the one type of E. coli that was resistant to antibiotic A, that particular gene can be cut by the plasmid that is introduced, and another gene which can be inserted in its place, which would work so that with the E. coli would be resistant to other antibiotic. So these were their experiments, and their labs were transferred from UC Berkeley to Stanford, and they were successfully able to carry out these experiments for gene editing. And so that was the start of genetic engineering. Okay, and Now, this idea was one of the investment guys, who was working with, I think, I don't remember the name of the company, but he was an investment, stock analyst basically, and he was like 29 years old. He was interested in reading some of these scientific journals, and he had read about this recombinant technology, and he thought about using commercial application. So he requested a meeting initially with Paul Berg, who had done the original works, about of plasmid and E. coli. But Bob Burke, was not interested in talking to him. But Herb Boyer did, talk to him, and from a 1 hour meeting, they decided to, commercialize and start a company. And so they each invested $500. Okay, and then they decided, okay, well, they would be able to use this technology to manufacture desired chemicals. Okay, and so this was the start of genomic technology or genetic engineering, as we'd said, and this was again, so the financial stakes that were available, and so this was the rise of genetics and the rise of, Wall Street getting involved, and they decided to form a company. Initially, they wanted to name it Herb and Bob, okay, right. So, but then again, they came up with a better name, which was called Genetic Engineering Technology, and the name was abbreviated as Genentech. Okay, and so that was the start. That was the big behemoth in biotechnology at the present time. Okay, and so initially, and again, just to go over this thing, as to how they decided to manufacture, initially some other chemicals, but then later on, the biggest need was for insulin, because originally insulin, which was discovered in the 30s, it came from pigs and cows, and it took a lot of carcasses and pancreases, to manufacture. And with the rise of diabetes, it was impossible to keep up, with the supply and demand. Okay, so they decided to settle on insulin as a molecule. Okay, so based on reverse transcriptase, they were able to make copies of DNA inserted inside the plasmid, and then the plasmid with the desired gene was introduced inside the E. coli, and then the E. coli merely went on making pure insulin. Okay, so this is, this where they obviously it was a lot more complicated. This was just in a laboratory, so they had to do it on an industrial scale. Okay, and so but this was the big molecule that they decided. Before that, they'd used smaller molecules as for test this thing, which is called somatostatin, which has something to do with growth hormone. Okay, and but again, so this was the big testing. Now at the same time, on the East Coast, other people were doing the same thing, but again they were using human cells and trying to synthesize. So they had to meet with their biosafety requirement. So there was a big race between the company that was on the East Coast, and also on the West Coast. And the Genentech had decided to take the individual nucleotides. Insulin is made of 2 chains of amino acids, and those 2 chains where these nucleotides were assembled, just like putting Legos together, basically. So that's how the insulin was synthesized. And they were able to, they were competing with the East Coast group that was working in, actual the cell line. Okay, and so that's one of the reasons why they could not work in Cambridge. They had to borrow time from a biohazard safety laboratory, in UK, and the UK rules were a lot more laid back, so that slowed them down. And somewhere along the line, they had their signals mixed up. So instead of synthesizing human insulin, they were synthesizing rat insulin. Okay, so eventually the race was won by Genentech, and eventually the company was bought out by with that contract with Lilly, who was the giant manufacturer of insulin. Okay, and that was the rise of, and that was so once the restrictions were lifted for more than dangerous direction, and the 2 big a commercial success that was placed with somatostatin, which has to do with your growth hormone and insulin. And so this were, and then Genentech, because it was a synthesized product. And so the Stanford listing that they were able to get a patent on it also about the technology, because they were not synthesizing a new gene, but they just went for the technology itself. so they were able to monetize it in a big way, so to speak. And obviously now all the insulin that you take now is synthesized, with recombinant technology. Okay. And so then Genentech, went public, back in 1980, and that was one of the big success of biotechnology. And these two people, that's Herb Bayer, and The Rise of Genetic Engineering, made on the cover of Time magazine. And for the current timing, it may not be a big this thing, but people who were, postdoc students, and they became multimillionaires very quickly. And obviously, Genentech has gone on to synthesize a lot more successful drugs, including, not after somatostatin, they devised, a factor 8, because again, remember, this is 1980. This was the same time where HIV was raising its ugly head, and the first case reports came out in 1981 of the so-called Haitian disease, or the gay disease, as it was labeled at that time, and then it was, and people who had hemophilia, they had to get the factor 8, which was not, the blood was not screened, and then and there was a kid in somewhere in Indiana who had HIV, and obviously the local people, local population did not want him to attend school, and he became a celebrity cause. People who were big, such as Elton John, went to bat for him, and a law was passed. I forget his name, type thing, but Mukherjee tells a story about how it was politicized type of thing. And so Genentech then worked on synthesizing factor 8. That was the next big hit, the synthesizing. So, and that avoided the risk of getting HIV from factor 8 transfusion. So that was the second big hit. The third big hit was something called tissue plasminogen activator. And that is, to put it in plain English, it is called clot buster, which dissolves the blood clot. Okay. And that revolutionized cardiology as we know it when tissue plasminogen came out. That revolutionized the treatment of heart attack, because we were able to dissolve the clot and buy time, and eventually open up the artery or send for bypass surgery or what have you. So those were the big success stories of Genentech. And obviously now, they're synthesizing a lot more drugs. And now at the same time, on the other side of the pond, the sequencing of, was done and was discovered by the crystallographer, par excellence, one of the unassuming guy who discovered it was early in the early days of crystallographer, and he had already won one Nobel Prize, and he had come up with the way of doing, okay, let me go back through this thing. Okay, this is a Fred Sanger, is that genetic analysis system about the DNA sequencing, that method that he had discovered basically about, various DNA templates were primered and had put in a stop codon at the end, and based on electrophoresis, because these have different molecular weight, how they migrated on gel, and with the electrical current, he was able to isolate and come up with a DNA sequence, okay, of a sample that was placed. So this was called the Sanger method of DNA or RNA analysis. So this was one of the earliest ways to be able to determine the sequence of the genetic code of whatever element that you're using, be it RNA or DNA type of thing. So he put in something called a stop codon, and based on how this electrophoresis pattern migrated with electrical current, okay, so the jet electrophoresis, so applying electrical current. And so this was, the groundbreaking discovery, and again, was widely utilized for determining the sequence of various organisms, starting from the lowly bacteria. And again, just as a digress, a physicist turned biologist, named Carl Woos at University of Champaign-Urbana discovered the whole new phyla called Archaea based on the Sanger method of remember we talked about the ribosome, and based on the molecular weight of ribosomal RNA, he was able to find out that there were not just two prokaryotes and eukaryotes, but there is a whole two kingdom, which was called Archaea, which were the original bacteria that he discovered in. And these are the extremophiles or thermophiles that can thrive in extremely hot, volcanic water in Yellowstone. And so he discovered the whole, but he does not get as much recognition as he deserves, but because he discovered changed the whole biology as we know it, and discovered the whole new class of organisms, that have played a huge role. Okay, and I may have mentioned as to how the one bacteria was swallowed and became mitochondria and started the evolutionary process altogether. Okay, and again, so this is, as we said earlier, that it's a lot more complicated in a more sophisticated way for those with more interest, how the DNA, gets down to messenger RNA. And this is the basic code that we talked about earlier, but again, things are a lot more complicated, especially in eukaryotes, because the DNA is, we showed about the intron and exon, but this is a more scientific thing, but we already went through that just to, okay. Yeah. Okay. And again, so there are two types, and most of the DNA is in inside the nucleus, but there is also a small DNA, which is a relatively small segment that is inside your mitochondria. Mitochondria are the powerhouses, type of thing. And interestingly, or some of you may know it, but I wouldn't assume that everybody, but that is inherited only from your mother's side. Okay. So, okay. And that, so we can trace the line. So this was and the origin of mitochondria was always a puzzle. And it may have been that in the early days of evolution, mitochondria were also bacteria that were engulfed by another bacteria and decided to say, well, this is a pretty good home, and not to, they lost some of their genes, and only the essential genes were survived. And that's how the one bacteria was engulfed by the other. And this mechanism of something, the biological term that we have to use is called symbiosis, that was proposed by a woman biologist named Lynn Margulis, who was married to Carl Sagan. Okay, and again, as it happens in biology with paradigm shifting, her idea was not initially accepted, but eventually, after about 10-15 years, when other people duplicated her work, she was proven to be right, and that may be the origin of mitochondria. And mitochondria are the power cells, and because we've talked about the messaging and the code and the information, but we have not addressed something called energy, because all this requires energy. That energy is done with something called, with photosynthesis and something called, and respiration in photosynthesis in plants, and with respiration in humans and mammals, Okay, and so this idea was discovered by a guy named Peter Mitchell, who was an English biologist, and his able assistant named Jennifer Moyle, and he established something called the proton gradient, and which is the way of how energy is utilized inside the cell. Okay, but that gets more complicated, and it's something called, which we had talked about in other classes, something called Krebs cycle, and it is the proton gradient that provides the energy, which is the workhorse called adenosine triphosphate, which is the source of energy inside the intracellular network. Okay, again, so that gets down to the story of patenting. Okay, genes, obviously, people learned to commercialize. And an Indian biochemist who worked for, I'm trying to think, who did he work for? And I think it was one of the oil companies, I think it worked for Exxon or something like that. I forget, And so he had discovered something called oil-eating bacteria. Oh, General Electric. I'm sorry, my notes are, is name. And so this was, and they wanted to patent the gene, and that case went all the way to Supreme Court and took almost 10 years, but eventually he did get the patent because it was something that was totally different, type of thing. So they were eventually granted patent type of thing. So, and again, the same patent thing, that was used, by Stanford and Genentech eventually, but what with the they actually patented the process of cloning. Basically, it was patented by Stanford in 1980, and again, so the TPA patent was also granted with Genentech. And the cases went on between the East Coast company called Genetic Institute and the Genentech on the west side. Okay, and and again, the different countries decided differently, because in US the Genentech was granted the patent. In UK it was not, because it was decided in front of expert judges instead of jury. Okay, and this again started the mad rush as to who was on the first, so to speak. Okay, and that controversy continued with varieties of procedures in biological sciences, especially with the PCR technology that came in later on, and even for the HIV blood test, the patenting, and so this was again started the arm race between the academic institutions, commercials, okay, and a variety of time. For a lot of these things, the patients actually lost out based on the cost, okay. And again, the varieties of essential drugs, the patents have, and in the other things, some of the technologies, the people in Europe were a lot more laid back about patenting. And I give the example in the medicine class, the Medical Research Council in UK, they failed to patent the technology of monoclonal antibodies. Okay. And they thought, oh, there's not much use for it. So even though that was discovered by them, they didn't get around to patenting it, but the Americans promptly did, and that just opened up the floodgates, and so they lost out. So, but now I think they've gotten wise eventually. Okay. And so this is again, the varieties of then again come of diagnosis. Obviously the low-hanging fruit, as it was diagnosing rare diseases, which are affected, and there's a whole rare disease consortium, which affect, the incidence varies, but there are as many as 7,000. So that is enough work based on varieties of, those earlier gene mapping technologies were, the sequencing was very time consuming, slow, and deliberate, and there was done with something, because the gene mapping, or other word was positional cloning. And it was literally like trying to find a needle in the haystack. And this is the famous Francis Collins, who posed in '89. He's the head of NIH, and just retired, within the last few years, okay, and and took over from Watson for the Human Genome Project. And CFTR is he discovered, with methodical gene mapping technology, cystic fibrosis gene. Okay, so these are, I'm not going to go through the litany of 7,000 genes, okay, but just go the important ones that you are all familiar with. This is the cystic fibrosis gene that was discovered by Francis Collins, and Mary King. She did based on family analysis, of people with breast cancer. So and she was a postdoc on the West Coast, and she discovered the BRCA gene, okay, in competition with a commercial outfit called Myriad Genomics that had the access to the data from the Mormon Church, from in Utah database. So there was a race between the two. And last but not the least, the third famous gene is the Huntington's disease. That was pioneer work was done in Venezuela by Nancy Wexler. Okay. And there was a saying as to cystic fibrosis was known in Europe for a long time. In fact, there is a saying in German, okay, and which I won't try to read the German, but it's on my slides, Woe to the child who tastes salty from a kiss on the brow, for he is cursed and soon must die. That was a saying in German in the Middle Ages. So cystic fibrosis was a lot more common in Northern Europe, and so that was one of the, like we can say the marker disease type of thing. So these are some of the diseases that, there are genes. So, okay. And these are the genes that I already alluded to about the genetic in the great clone or die. So they first undertook the small hormone called somatostatin, which is inhibitor of the growth hormone. Okay. Then this 1978 insulin. You can see this was a relatively easy work. 14 amino acid. Insulin was 53. But the big one was 1983 factor VIII. You can see how complex the task was. That to clone as many as 2,350 amino acids. And then later on, the TPA, which I talked about earlier, tissue plasminogen activator. Okay, and that is a lot to do with hemophilia, HIV, which we already alluded to. Okay, and babies who died, from transfusion. Okay, all right, and again, and then insulin was again, this was based on anatomy. People had already diagnosed something called islet of Langerhans, and the enzyme that came out of islet was called isletin, ISLETIN, and that later on morphed into insulin. Okay, and so this was one of the earlier ones. Okay, all right, okay, and this we already talked about this in cloning technology that we talked about. It's a very elaborate step. We can try going through that. Okay, genetic cloning. Okay, you can see it is cleaved with restriction enzymes. Okay, and the phage DNA. Okay, I already showed that earlier, how the phage is, just like a base, a DNA molecule with a needle type of thing that infects the bacteria, that is transferred and released on denatured DNA that is synthesized with other factor VIII. Then it goes on. They make copies of DNA with cloning method. Again, more enzymes are used. Okay, and then grown again finally. Okay, so this is how, and this is done on industrial scale to manufacture type of thing. Okay. Okay, now at the same time, with the genetic technology, we'll just have a couple of more slides, then we can stop probably. Okay. At the same time, the thing was two promises of genomic technology were one, to cure diseases, and second was to grow food. And the things that were going about, that the big thing that was done at the same time was to avoid the, because Monsanto was under pretty bad name because they had synthesized DNA, various insecticides, and they were considered Agent Orange, so they were not exactly popular in the general listing. And they had the good idea of trying to get out of chemicals, and this is when they came up with a, natural, and it was already used by other farmers called Bacillus thuringiensis. That's a bacteria that plants expressing that, would reduce the need for and prevented that from insect infestation. Okay. So this was, a way to introduce the gene inside the plants with something called gene gun. Okay, so that is called the gold-coated particles that would insert the gene inside the bacteria, okay, inside the plant, and would avoid, having to use insecticide. Okay, and again, so but they marketed it rather aggressively, and instead of, farmers were just basically renting seeds, and so that started with, there was something about the food that people in Europe, for whatever reasons, again, at the same time, we said that the political climate was that the food shortage and the mad cow disease was around, and so that, that created a problem and gave the bad name to Monsanto against, and started all the protest against GMOs. Okay, Bacillus thuringiensis. Okay. And so, and there are varieties, but that's a lot more acceptance in US and the rest of the world about GMOs, and this is the example of the gene guns. and it has, a big plus in reducing the, of food scarcity. Okay. Okay, and again, there's the this thing that has been testing with the golden rice, which is, vitamin A introduced inside rice. And again, but the Greenpeace has, for whatever reasons, they made their big cause in protesting. And, these are the examples of the countries that do accept GMOs, I suppose not. And one scientist, he finally had, a realization that he was wrong. And he actually wrote a book as to why we had it so wrong about GMOs. Okay. All right, so I think it's coming up to 3 o'clock, so we should probably stop here at this stage. Okay, I will stop the recording and I will continue some of the step, next time. Okay, let me pause the recording or stop recording rather.